All right, everybody, we're going to go ahead and get started. Thank you so much for being here. My name is Cinnamon Moffett, and I'm the Research Program Manager here at Oregon State University's Hatfield Marine Science Center, and I welcome you to today's seminar. This is a hybrid event, so we have folks online and folks in the room. For folks that are online, Roseanne is our volunteer that is going to be monitoring your questions in chat. Your cameras, mics, and screen shares have been disabled, but we hope that you interact with us in that chat box, and Roseanne will read out any questions that you put in there to today's speaker. For the folks in the room, I'm looking around. Most of you have done this with me before. Just raise your hand if you have a question at the end, and I'll bring the mic around to you so you can ask your question so folks online can hear you as well. There's also a mic stand in the back corner if you'd like to use that. Wanted to make a couple of quick announcements. We are already in February, which is amazing, but next week's seminar is Angela Skorka, who is with the Marine Mammal Bioacoustics Ecology Lab with the Marine Mammal Institute here at Hatfield. She's going to be doing a talk about exploring the drivers of whale migration from the tropics to the poles. Been trying to get her here for a little while to give a talk, so I'm very excited that she's going to be here next week. Also just wanted to remind folks that our next Science on Tap is next week on February 7th, where we have Suzanne Brander will be here talking to us about plastics impact on organisms in Oregon and beyond. That was a talk that was scheduled during the winter and we had to reschedule it. So Suzanne Brander is going to be able to give her talk. It'll be in this room and online starting at 6 p.m. on February 7th. But the reason that everybody's here and online today is for today's speaker, when we're very excited to have Dr. Kelly Sutherland with us. She has been an associate professor of biology at the University of Oregon's Oregon Institute of Marine Biology since 2012. I'm looking at her because I'm pulled this line off of online, and so I just want to make sure that I'm correct. Been there since 2012. She has a bachelor's from Tufts, a bachelor's from the University of South Alabama, and a PhD from MIT Woods Hole Infographic Institute. Her lab works at the intersection of biology and physics, exploring the interactions of marine organisms with fluid environments. And she's here today to talk to us about her work. Some of you might know her. She's a figure that comes around and goes out to sea with several of the folks in the room. So we are excited to learn a little bit more about her work. I'm going to hand it off to Dr. Kelly Sutherland. Go ahead.
Thank you. I think I need to turn this mic on. Okay, how's that? Great. So good to be here in this really beautiful space. This is my first time in the new building. So really, such an inspiring place to be. So you may have gathered from this cover image here that we study zooplankton in my lab, and primarily we work on gelatinous zooplankton. And before I get any further, let's see if I can actually advance this slide. Okay, passed the first test. I always like to thank the folks in my lab group at the very beginning. I'll acknowledge the current members that are in my lab, Tara, Alejandro, Farzana, and Elizabeth, who are currently in the lab. And I also want to acknowledge some of the close collaborators that I've been working with the last few years, including, in particular, Sue and Bob, who are here at Hatfield, and also Rick Broder, who's Hatfield-based. So digging into gelatinous zooplankton a little bit, the interesting thing about this group is that it's not a taxonomic group, it's more of a functional group. It turns out that almost every single marine phylum has gelatinous members, right? There are gelatinous mollusks, gelatinous worms. We've worked on in my lab ctenophores, cnidarians, pelagic tunicates, which are actually chordates in the same phylum with us. And the thing that they all have in common in this operational group is they have these large watery bodies. And this allows them to put on size really quickly when conditions are optimal. It allows them to have large capture surfaces for capturing food in a dilute environment. The other thing they all have in common is that historically they've been understudied. And the reason for that is that they're fragile, their tissues tend to fall apart when we use traditional sampling techniques. So one of the things we've tried to do in my lab is just fill some gaps and also to develop new technologies to study these fragile organisms. So how do we study gelatinous zooplankton? One thing we do, and I'm going to be showcasing some wonderful images from Mark Farley, who most of you probably know. He came out to sea with us. So Sue, Bob, and I have had 2 NSF grants together now, and we've gone to sea together a bunch. And this is one of the ways that we study gelatinous organisms. We go out to sea and we use traditional net sampling techniques. And when we do that, and here's some more familiar Spinagle Cowan folks, Miriam and Jamie are spraying down a sample into a cotton. And when we do that, we get jellies back sometimes, but they look kind of like that. So we can ID, that's a salp. We can see what it is. We can probably measure it. We can maybe do some biogeochemical studies with it, but we certainly couldn't do any experiments or behavioral studies or anything like that. So there are certainly limitations to using traditional net sampling. And I was really thrilled when Bob and Sue got here and brought ISIS with them, so that in the Northern California current, we can now get a better handle on the distribution and abundance of gelatinous organisms. So together we've been working on food webs and also plankton size spectra in the Northern California current. And I'll just underscore how amazing it is, the disconnect between what comes up in the nets versus what we see on ISIS. So one player I've noticed are Bulinopsis, the lobate ctenophore. We see tons of them in the ISIS images, and virtually none of them come up in the nets. They're just going right through the mesh. So if you study copepods, they're probably getting eaten in huge numbers by these lobate ctenophores, and we just have no record of them. And there's a lot of gaps like that in our understanding of, in this system, which plankton are here, but also in other marine systems, in other places. The other way that we study gelatinous plankton in my lab is to kind of take the laboratory underwater. So we use blue water scuba techniques to do more organismal scale studies of predator-prey interactions. And we also work on locomotory mechanics. And a lot of times we're developing new camera systems, kind of bespoke camera systems, in order to study these organisms. And then finally, when we are diving, we're often collecting specimens that we bring back into the lab. One of our interests is how marine organisms interact with the fluid in and around themselves. So this is an example of a particle image velocimetry video. This is an obelia, so very common hydromedusae. You have them here on your docks. It's about 1 millimeter in diameter. Here we were interested in understanding the fluid flows in and around the tentacles and how that relates to prey capture. But I just wanted to showcase that this is a technique we use a lot, where you're seeing velocity vectors and vorticity contours. So we do those experiments in the lab typically that are a bit more quantitative. For today's talk, so I mentioned we kind of work across the gelatinous organisms, but I want to focus a little bit on the pelagic tunicates for today's talk, which in and of itself is a really diverse group. So in this group, we have the salps, doliolids. Let me see if I can get a pointer going here. Doliolids, the pyrosomes, and appendicularians. And all of these organisms are using mucus meshes to capture their prey. And they're often, with larvaceans in particular, they're often second most abundant to copepods in coastal systems. And these mucus meshes, an interesting thing about them is that the size scales are very small, so around a micron in size. And so they're capturing things that are many orders of magnitude smaller than themselves, down to even viruses. So there's evidence that appendicularians can actually concentrate viruses in their mucus meshes. And just to kind of show that in plot form, this is a plot that shows pelagic tunicates, but we're also including pteropods, and kind of thinking of this group as a group that uses mucus mesh filters to concentrate small prey. And if we look at all these mucus mesh grazers and we consider their prey to predator length on the x-axis there, and then we also consider their clearance rates on the y-axis, you'll notice that not only is there a much greater ratio between predator and prey for these mucus mesh grazers, but they also have higher individual grazing rates than some of these other common grazers in marine systems. So things like copepods and ciliates. And where are we doing this work? So I'll share a couple different stories, but a lot of the work that we've been doing recently is out in the oligotrophic open ocean in the North Pacific Gyre. So during COVID, we started doing some work off the Kona coast of the Big Island. So that's where this image is from. And it's relatively new for me to work in the Hawaiian Islands. And I find it very novel that we're just right in the middle of the gyre. And when we go offshore on dive boats, we see this amazing diversity of oceanic plankton. We've found almost every single salp species over time diving offshore there. So it's been a really productive field site for us. And I mentioned that pelagic tunicates in particular are eating these really tiny cells. And so just to kind of remind all of us, the open ocean is really dominated by small cells. So this is an image from the MIT Darwin project. And to really like drive things home about kind of the importance of these mucus mesh feeders. So they're eating these really small things. And we often talk about them in terms of short-circuiting the microbial loop. So they're eating viruses, they're eating tiny phytoplankton, they're eating bacteria, and they're pretty large in size, and they become prey for much larger things like fish in some cases. They also, many of them have, like salps, have very fast sinking fecal pellets, and so they're responsible for high amounts of flux. And we don't have to go too far to find pelagic tunicates. We have them right off of our coast. Many of you probably are aware of let's get this video playing. Maybe I can't do the laser pointer and click on videos at the same time. Okay. So with the warm water blob, we had these huge looms of pyrosomes showing up here off the Oregon coast and all the way up into Southeast Alaska. And in a way, I was pretty thrilled because I'm someone who studies pelagic tunicates, and here they are showing up on my doorstep in Oregon. So that was actually pretty kind of opportunistic. So we've been able to do some work on pyrosomes. This is, you're looking at a video from a camera that was on a CTD that was being lowered. And you'll notice as it kind of goes through the video that there are definitely these dense layers of pyrosomes. And we were able to characterize a little bit their distribution with depth. So pyrosomes aren't the only bloom-forming pelagic tunicates. Many of them are capable of forming these large blooms that in some parts of the world are increasing in frequency and in magnitude. Meanwhile, there's been this ongoing paradigm that feeding by pelagic tunicates is non-selective. So this idea that they're just hoovering up seawater and associated food particles and kind of collecting whatever sticks in their mesh. So sort of like the spaghetti strainer model. And this paradigm has really persisted. Just in this past week, I ran across a paper that I haven't had a chance to read about Salpa thompsoni, which is a common salp near Antarctica, that called it the non-selective grazer, Salpa thompsoni. So I was like, okay, we have more work to do to show that these organisms are probably very selective in their feeding. So what I'm about to do is share some evidence with you. This has been a question that's been driving some of our research. Do pelagic tunicates feed selectively, or are they just passive vacuum cleaners? And we've also addressed some of the underlying mechanisms that give rise to that selective feeding. And I won't talk too much about the mechanisms, but we've done a lot of experimental work with polystyrene microspheres to get at some of the selection mechanisms. So if you invite me back, I can talk about that some other time. But kind of keeping on with the pyrosomes, so we had all these pyrosomes showing up off the Oregon coast, and working with Dr. Ann Thompson, who's at Portland State University. She's a marine microbial ecologist, and I should mention that she and I were in the same graduate program together in the MIT HUI joint program. And so I was really thrilled when she arrived in Portland. And for those of you that are listening that are graduate students, you never know who's sitting next to you. You might one day write an NSF proposal together with. So together with Anne, and Anne joined some of the cruises with Bob and Sue's group. We looked at background seawater and pyrosomes and just compared the two. So without diving too deep into this, the samples shown on the right side of this plot here are amplicon sequence variants from the seawater. And then on the other side are the pyrosomes. So there's a lot of detail on this plot, but it's pretty clear that the two things are different. The seawater and the pyrosomes look different. One of the complications with pyrosomes is that they have very tiny guts. And so in this study, we've talked about the ASVs as being the microbiome of the pyrosomes. And we were able to kind of disentangle which things we thought were probably putative prey versus associates. But that was something that was a little bit challenging to disentangle. But one thing that we did notice was that it seemed like there were diatoms in higher abundances in the pyrosomes relative to the background seawater. And for sure, there are certain things that are in the seawater that they aren't collecting in their mucus meshes. And it's always nice to see things with your own eyes. And so in my lab, we did some work using environmental SEM to actually image some of the gut contents. And we saw some of the same things showing up in these images that we had seen from the sequencing work. So things like centric diatoms and also pennates. I'm just looking at the pictures and reminding myself. So shifting to a different system, so that was off the Oregon coast with pyrosomes, but now shifting to more oligotrophic systems. We've done similar work. So again, working with Ann Thompson, we've been doing some work off the coast of Florida, working off of West Palm Beach. And from there, we can get into the Gulf Stream waters pretty quickly. So in this case, we were looking at a different set of mucus mesh grazers. So we were looking at the salp, Iasis cylindrica, and a pteropod, corolla. So corolla makes this big mucus bubble that it sediments prey on. And it's a similar story here where I'll get my pointer going again. In the center here are our seawater samples. And in this case, we're looking at just relative proportions of different potential prey types. And in this case, we've kind of narrowed it down to things that we think are potential prey that are in the guts. We were able to dissect out the guts in this case, because they have very clear, in each case, the salp and the pteropod has a very clear kind of nucleus that you can dissect out. And there's certain items like Prochlorococcus that are really common in the seawater, but not showing up in salp guts over here or in pteropod guts. We had a pretty low sample size with the pteropods, but we thought this was some pretty interesting evidence that feeding is not just based on size alone. So if we go over to this plot on the right here, so this is for the salps where we had a decent sample size. We have prey types here on the x-axis, and we also considered the size of these different prey types. And Synechococcus was in a very high proportion relative to the background seawater. And it's a very small cell. It's on the order of a micron. But Prochlorococcus seemed to be negatively selected for. So two cells that are kind of similar in size, they're not the exact same size, but there definitely seems to be some selection going on. So the next thing that we're doing with this work together with Anne is incubations. So in each of those studies that I just showed you, we took seawater and we took guts and we compared them. And what we're not getting at are rates of removal. And so we're currently doing some work this is now off the Kona coast of Hawaii, where we collect salps while we're diving. So they don't even know they've been collected. We capture them in a jar, and we have these little injection ports on the top of the jar. So then we can use syringes to take seawater samples through time and look at compared to a time zero sample jar that we collect, we can see how much prey are being removed This is Tara Hubert, and who's currently in my lab, and Kevin Duclos, who is a former postdoc doing these incubations on board a dive boat there. So we're still working on this problem of selectivity, but there's certainly mounting evidence that feeding is selective, and this is from a review paper that Anne and I wrote together, just trying to get at what we know from the literature at this point. And if we look at the different pelagic tunicates, they have there's differences in clearance rates. There's also differences in which prey types that they're consuming. So these are different prey types, and I'm also showing the size of the prey types here, and who's eating what. So there's a kind of a rich story that we're trying to disentangle here. So to be continued on the exact mechanisms, but it's clear at this point that grazing is selective. It's not just this vacuum cleaner model. And the reason that this matters is that when we situate these organisms in food webs, they're abundant, even though our estimates, we're way underestimating how many of them are out there. But they are abundant, and they are consumed by things like fishes, larger jellies, seabirds, and they also have this property where they're responsible for depositing fast sinking carbon packets to the seafloor. So sometimes that's in the form of dead jellies, sometimes it's in the form of fecal pellets. In the case of appendicularians, they've got these mucus houses that they're discarding, sometimes as many as 15 a day. So if they're removing microbes at different rates, if they're removing them non-uniformly, then we need to know about that if we're going to understand how carbon is moving through marine systems. So I've been talking a bit about the grazing and selectivity. I want to move to thinking about how many of these things are out there. So getting back to the pyrosome story and kind of underscoring that that these organisms are distributed everywhere, and they can be quite abundant. So when the pyrosomes showed up here offshore, we wanted to know where they were. So how abundant are they? How are their numbers changing over time? We're still kind of trying to figure this out. So Elizabeth Daly is here in the audience, and Rick Broder has been spearheading this effort to kind of bring together a lot of different investigators that have worked on pyrosomes, and trying to get a bigger picture view all up and down the coast of how their numbers have shifted over time and what the environmental drivers might be. So it's amazing how, like, when the public asks these questions about why are the pyrosomes here, like what are they doing? It seems like a reasonable question, but we still don't really know. I mean, we're I shouldn't say we don't know. We're gaining some insight and some knowledge, but it just takes time to disentangle a question like that. So one of the things that a former student, Hillary Sorensen, worked on, she actually went out on some NOAA cruises with Rick, and just using a combination of net tows and also cameras mounted on CTDs. She just measured the abundance at these different stations and showed that they're not uniformly distributed, and there seemed to be more offshore. This happened to be in 2017 that she did this work. And then after that, another student, Joanna Lyle, was interested in this problem, and she used some of the same data from a different set of cruises to look at vertical distributions. So when we mount cameras on CTDs, or if we use mock nest tows, then we can look at distribution with depth. And I'm showing a couple images, the types of images that she analyzed from the mounted cameras. And from the mounted cameras, you get more fine scale distributions. And then she was able to pair that with the bars, which are samples from the mock nest. And then on the right, we're looking at a profile of temperature and fluorescence. And I'm just showing you one example plot She has a bunch of plots like this from a bunch of different stations, but the pattern that we saw was that these layers of pyrosomes were hanging out where the chlorophyll was at a maximum. So they're taking advantage of these layers of phytoplankton, most likely, and grazing there. The other thing about their distribution is that it's not uniform. They engage in diurnal vertical migration. This has been shown before in other parts of the world. But she was able to show with the same setup that during the nighttime, they tend to be distributed up near the surface, and during the daytime, they are distributed more throughout the water column. So pyrosomes are one example of a zooplankton that migrates, that engages in this vertical migration. They're certainly not the only organism that does this. Every single night, we have this huge migration, right? That's where a huge amount of biomass is coming up to the surface and down to depth. And I'm kind of using this as a bridge, this slide together with the next slide, to move into another area of research in my lab. So we've been talking about pelagic tunicates and their feeding and their potential predation impact. Many of them are engaging in these impressive vertical migrations, and that leads me to another area of interest, and that's thinking about their locomotion and their locomotory mechanics. So another area of research that we're really interested in is understanding how plankton swim. And our focus has been on jet-propelled organisms. So we see a lot of jet-propelled animals in the plankton. It seems to be an effective strategy. It shows up a lot in vertical migrators, actually. And for a while, together with a number of collaborators, we were really focused on cnidarian medusae as a model for understanding locomotory mechanics. And interestingly, a colleague of mine, Brad Gemmel, did this study where he compared jellyfish with a bunch of other movers. So not just swimmers, but also flyers and terrestrial organisms. And so that plot there shows body mass on the x-axis and net cost of transport on the y-axis. And Aurelia aurita, the moon jellyfish, has the lowest cost of transport. So arguably, jellyfish are the most efficient swimmers on the planet. And that is due to a lot of really novel fluid mechanics that, through a number of different studies, this has been a large set of collaborators. Together, we've unpacked some of the different fluid mechanical phenomena that give rise to this really hydrodynamically and energetically efficient swimming. So we kind of wrapped up over a decade of work in a review paper about that a couple of years ago. And what I want to spend most of my time talking about is actually multiple jets. So jellyfish swim with one jet. What happens when you string a bunch of jet-propelled organisms together and coordinate those jets? So we've been really interested in jet propulsion by salps, which have this colonial life history stage, where you have a bunch of zooids that are strung together. And we've also done work on siphonophores, but I'm going to stay with the pelagic tunicate theme, so I'll talk about the salps today. So this is a very linear species of salp, Metcalfina, that we took video of off the Kona coast of the Big Island of Hawaii. So some of the questions we've been interested here are understanding they're basically form and function questions, right? So what are the body shapes, the kinematics, and the fluid mechanics that give rise to effective swimming in some of these multi-jetters? And it gets pretty interesting with salps because they have this huge diversity of colony structures. So it's a really fun design space to think about. So you've got this kind of two levels of organization where you have differences in the morphologies of the zooids, but then there's the secondary architecture in terms of how the zooids fit together. So there's these clusters, there's whorls, there's these linear chains. And a lot of times when people study salps, they just bring them, they come up in a net and they're on the deck of the boat and they come apart. And so we've really put a lot of effort into studying these organisms in the natural environment to understand their swimming. And I'll just highlight that a postdoc in the lab, Alejandro, has really been spearheading a lot of this work. This is just kind of the line drawing version of these different morphologies that we see. And actually, this goes back to, I'm just reminded, this goes back to work that Larry Maiden did, who was my PhD advisor at Woods Hole. Okay, so Alejandro's been working on this problem, and he has a background in thinking about phylogenetic relationships, and so he was really excited first just to kind of map out the phylogenetic relationships. So he's been doing some work with that and thinking about how during development we get these different chain structures. So if you look at solitary salps that have a developing chain, they all start off looking like this transverse arrangement, which is the zooids that are side by side. So Pagea confederata, for folks that are familiar with salps, is kind of the classic transversal chain. And there's a number of different transitions that give rise to the different chain morphologies that we observe across species of salps. So I'm kind of creating this design space, but I now want to drill down into one species in particular, just as a way to kind of show you how we think through a problem and kind of map out how organisms work, really, in this case. So I'm going to focus in on a species called Iasis cylindrica that used to be called Wheelia cylindrica. Its name has changed a few times. And so this is a very streamlined linear chain. And the video that you're seeing is a high-speed video that's been slowed down, just so you can kind of see how the swimming mechanics work. So fluid is getting pulled in through the front end, through the inlet side of the zooid, then circular muscle bands contract, and we have fluid ejected out the back end. And you can also see this very sweet little developing embryo that will become a solitary salp. So we got interested in Iasis because when we were diving first off of West Palm Beach in the Gulf Stream, we noticed that especially the long chains swim in a helix. And so we were just really curious about this. What's going on here? Is this, why is it swimming in a helix? Is there some adaptive advantage of swimming in a helix? And so we just kind of started to dig down into this problem. And because this is really a three-dimensional problem, these animals the chains are 3D and they're moving through 3D space, we really needed to use 3D methods to get at this. And so one of my COVID projects was setting up this stereo camera system that I was thinking about it when I came down here. Is actually inspired by some of the work that folks at ODF&W were doing with stereo camera systems. So we had the folks at Sexton, who maybe some of you have worked with, build underwater housings for us. And we calibrate the cameras with this calibration cube. This is that's kind of the glamour shot of the stereo system. And from these stereo images, we can then track the motion of these chains in three-dimensional space. And so this is some data that show the different morphologies of the whole chain. We noticed that the shorter chains were actually not swimming in a helix, they were spinning. So I'm kind of looking at this plot over here. So depending on the number of zooids, we saw different behaviors of the chains. So only the long chains that had, say, 30 or more zooids were swimming in a full helix. And it was really only with these three-dimensional techniques that we could kind of get at this. And then the other thing that we wanted to do is really characterize the three-dimensional shape of the chains. And so I'm showcasing some work from Kevin Duclos, a former postdoc, where he scanned a laser through a tank that had a salp chain in it that was hopefully not moving for a few seconds. So to do this, we collected salps in jars. We collected a bunch of seawater. And then Kevin actually had a tank built that was just sized to fit in a pelican case, so we could take it on the plane to Hawaii and have a large volume. Most jellies like to have a large volume of seawater. And so using this setup, we could get these three-dimensional scans pretty quickly after we'd collected the chains, and they were still pretty happy. And so the images up here just show the different views of the chain, and then when you put them all together, you can really see the orientation of the zooids. They're kind of at this slight oblique angle relative to the axis of the chain. So the fact that there's a little bit of this offset is likely what starts to create some instability as the chain gets longer and longer, and eventually you get this kind of buckling. But even with the shorter chains that are spinning, there's this offset is also responsible for creating a little bit of torque that likely leads to the spinning in the shorter chains. So that was the three-dimensional morphology. We also wanted to get at the kinematics, so how the animals are moving. So to do that, we use some bright field videography. So this is where the light is shining straight into the camera, and it works really well for jellies because you can see a lot of detailed structure. And during jetting, we see that there's a really low siphon angle, and it gets even lower. So that's what this plot is showing. The siphon angle gets down to, say, 20 degrees during jetting relative to the chain axis. So we can kind of corroborate what's going on. Did I get rid of my laser pointer again? We can corroborate what's going on with the kinematics by then looking at the jet wakes that are created. So this is maybe kind of cringy to watch. It's even cringier to try to get these videos. It's really hard when you have an animal that's swimming, a camera operator that's swimming, and then a person with a dye, with a pipette injecting fluorescein dye. We do a lot, like we squirt a lot of dye at salps in order to get a decent shot. And it does give us a nice, when we finally get a shot, here's a frame grab here. You can see, again, that the jet angles are very low relative to the axis of the chain. This method is, it's nice to be able to work with the animals undisturbed in the natural environment. We do this during night dives so that we get nice contrast with the background. We've also been able to get a few shots in the lab with the animals cooperate for a little while that are a little bit more quantitative. So here you're looking at another particle image velocimetry sequence. So this is where we use a laser sheet to illuminate neutrally buoyant particles that we seed into the tank. And then we can track the motion of those particles to see where the fluid is going in and around the zooid. So there are these really low angle jet wakes. Again, you can see with that are quite laminar as well, relatively speaking. So we've been talking for a while about a linear species, and so one of these linear species, and kind of unpacking the swimming mechanics. But I wanted to remind you that we've been trying to look at, it's kind of a daunting task. We've been trying to look at the whole diversity of salps with their different colony architectures and understand the mechanics that underpin their swimming. I should also mention it's usually these linear forms that we see doing these impressive vertical migrations, the equivalent of us running a marathon every day in terms of the distances that they traverse. In addition to looking at their swimming speed and the hydrodynamics of their jet wakes and their kinematics, we're also doing some metabolic measurements. So Alejandro's been doing work with freshly collected salps where they have, we use these oxygen sensors they're little sticky things that we put on the inside of the jar, and then we collect the animals. And so then he can start taking oxygen measurements right when they get on the deck of the boat. And again, it's just tricky working with these animals that they don't really like being in jars and in tanks. And so if we can't do the work in situ, then we're trying to do it as soon as possible after we collect the organisms. So he's been looking at oxygen, at metabolic rates with the oxygen measurements. And if you know the volume of the animals and you know how fast they swim, and we get the swim speeds from the field, then we can start to look at cost of transport. So how much energy is consumed per distance traveled, and starting to look at across these different chain morphologies. And it looks like the linear forms, maybe not surprisingly, are not only are they the fastest, so when we just look at how fast they're moving, they're the fastest swimming, and they also have the lowest cost of transport. And so it probably doesn't surprise you that these tend to be the species that are vertically migrating, whereas some of the other forms, like some of the cyclosalpas that have these whorls, or Pagea that has the transverse chains, they are slow and inefficient. But with salps, swimming and feeding are one and the same. So as they're swimming, they're also pulling fluid through their mucus mesh and capturing prey. So we think that the slow swimming forms are mostly just pumping fluid through their mucus mesh. And as long as they move enough to get into new patches of seawater, then they're probably doing fine. So Alejandro is continuing to work, and it's one of those studies that's just like very messy. Like we have to go out on a lot of dives to see enough salps of all the different species and kind of get the sample sizes. So it's been slowly coming together, and I'm really excited to kind of see everything come together in a full story. So I just wanted to kind of remind you of where we've been, and kind of underscore the approaches that we like to use in my lab group, where we're often working from the scale of individual organisms all the way up to ecosystems. So I was trained as a biological oceanographer, but now I'm in a marine biology lab, and I think of myself as someone who kind of sits at that interface, where I'm really interested in individual organisms, but I'm always thinking about them in the oceanographic context. And that's kind of reflected in the studies that we do, where we're kind of going from functional morphology, thinking about how things move and swim and feed, and getting all the way up into ecological function in a larger ecosystem context. And when it comes to the pelagic tunicates in particular, there's really interesting implications, because as I mentioned before, they're really widespread, but we're still getting a handle on how many of them are there. And they're very effective filter feeders, and they're capturing these really tiny particles that they package up and sometimes deposit to depth. And they're not grazing uniformly, right? They're selecting certain particles. And then some of them are also performing these impressive vertical migrations and bringing a lot of this material to depth. So I think I will end there. And I always like to end in my happy place, which is on a blue water dive. And I think that we probably have time for some questions.
That's a good happy spot. Thank you. All right, so we're going to open up for questions. I'm actually going to start and see if there's any questions online. Okay, if you have questions for folks online, go ahead and put them in the chat so that Roseanne can read them out, but I see questions in the room.
Could you elaborate on how the mucus is selectively feeding on the prey? I mean, how does that work?
Yeah, that's a really good question. So what's the selection mechanism? So there's I have answers to that question, but I could probably talk for quite a long time. And it depends a little bit on which organism you're talking about. So maybe I'll start with salps. So in the case of salps, and this is actually true across mucus mesh grazers, but where we've done the work is with salps. You have, instead of just simple sieving, where things that are larger than the mesh size, the holes in the mesh get caught, you also have other filtration mechanisms, including direct interception, where particles just impact the mucus fibers directly. And you can, with really small things, you can also have them just encountering the mucus fiber via a random walk with really small bacteria. Because there are so many more small particles than large particles in the ocean, you actually end up with a lot of particles just adhering to the mesh via direct interception on the mesh fibers. And so that accounts for some of the smaller particles being selected. The other mechanism at play is particle shape. So most bacteria are not spheres, they're rods. The majority of them are rods. And we've done some experiments with appendicularians comparing sphere and rod-shaped polystyrene microspheres. And we found that the minimum dimension of the particle is what matters in terms of prey capture. The other thing that we're experimenting with now that I'm really excited about is surface properties. So it's very small scales, things like the surface charge and the wettability of the particles and how they interact with the mucus probably matters. Some things are going to adhere and some things are not. When it comes to, so now switching to appendicularians in particular, they have this really fascinating and complex feeding mechanism where they have a bunch of different sets of meshes. So they have an external mucus house, and they also have an internal mucus filter inside the animal. And the selection gets really interested in appendicularians because they have these filters on the outside that exclude big particles. But then once particles make it in, they get attached to the food concentrating filter. And then they have to get unstuck from the food concentrating filter in order to go into they kind of go into this like straw-like structure, mucus structure, into the pharynx. And so there's a bunch of really interesting dynamics with sticking and unsticking. And we've done some work on that, looking at the pumping dynamics. So they do this pumping, and then they pressurize the house, and then they depressurize it. And then when they repressurize it again, particles start popping off the mucus. So there's a bunch of crazy dynamics going on that are sort of, they're passive in the sense that they're all kind of mechanical dynamics. So it's not that when we think of selection, sometimes we think of like an animal, like a fish or something that has vision, like picking things off. But there are all these kind of mechanical passive mechanisms that can lead to selection. So yeah, I could go on, but yeah, a lot of interesting and more work to be done with that, with understanding the mechanisms.
Great. Roseanne, question online. Looks like not yet. Okay, question in the room. All right, I'll do my best to run around and get you.
Back in the box.
Well, given that some salps swim at different speeds than others, and you said that the ones that are less efficient swimmers are probably feeding more, and I don't think these guys have many predators to avoid, so would you say they're probably just swimming to keep up with the vertical migration of their food? But I was wondering if there's any relationship between the habitat that they're living in and the swimming speeds that they need to maintain. Are there any relationships between like the overall abundance of food and the swimming speed maybe, or oceanographic factors like current strength?
Mm-hmm. Yeah, that's a really good question, because you see, it's kind of the classic paradox of the plankton. So you see this huge diversity in salps, and they're even doing different things behaviorally. So what's going on? Are they eating different things? It's a great question that we don't really have an answer to. Like, why do they really need to vertically migrate? It seems that the linear structure, the linear form has evolved multiple times, which is kind of interesting. And they do have predators. So there are, I mean, we tend to underestimate predation on gelatinous zooplankton because they don't have hard parts, they get digested really quickly, but a lot of fish are eating jellies. Leatherback sea turtles and mola mola are exclusive, almost exclusive jelly eaters. So they probably have more predators than we realize. So there might be an advantage there. The other thing we think about with the vertical migration is it could be for maximizing reproductive success. So it turns a three-dimensional problem of finding a mate into a two-dimensional problem. At least everybody's in one layer. But what's going on with the ones that don't vertically migrate? Yeah, these are questions. These are great questions that that I've spent a lot of time thinking about. That it's hard to design an experiment to get at them. But I think a really interesting question.
So first, I just want to thank you. It's been a privilege to be here to listen to an expert like you. And I like the real humble way in which you say you're still trying to quantify the relative abundance. But in the context of, I mean, you are an expert with jelly, so I'm going to try and pin you down there. I read once, for example, that, copepods are maybe the most abundant species and ancient species on Earth of today. How would you respond to that kind of broader description of your jellies in that kind of context?
Comparing them to copepods?
Well, I mean, I guess I'm just intrigued as to what, as an expert, what do you feel their overall role is in our way in which our ocean works? I mean, I was just amazed at how, how the parasomes expansion that we all experienced, it just took us all by so much surprise, but they were so present. Yeah.
So it's a good question. That's a really big question. Like, what are they doing? The other thing that I'll mention is that people tend to lump gelatinous zooplankton together, but they're doing very different things, and they're consuming different things, and they have different life history strategies. So it's hard to talk about them as one group, but it's clear, it's like crystal clear to me that our impression of how material moves through food webs, how predator and prey interact in the ocean, is missing some vital components. And some of it's from quantitative data. So when we look at things like the ISIS imagery, we see like, wow, there's a lot more jellies than are being estimated from net tows. But even from a qualitative perspective, every single time I go on a blue water dive, jump in the water, and every direction 360 degrees, there's jellies. And a lot of them have pretty full guts, even in really oligotrophic, so low nutrient waters, they have copepods in their guts. And so I'm quite sure that we are underestimating not only their abundance, but also because we're underestimating their abundance, we're underestimating their relative impact. So I'm giving you a little bit of a squishy answer, but they're squishy animals, and it's a big question.
All right, Roseanne, do you have a question online?
Yeah, super good previous answer, and this is the same question kind of, but any idea whether the size selection is optimal, more calories / M3 or biomass food?
Yeah, so is the size selection optimal? Yeah, interesting question. So big stuff is going to be better, right? So more calories if you eat something bigger. One thing that we've thought about is, like, every time we look at a salp fecal pellet, there's a lot of undigested material. So it may be that bigger things aren't completely digested. So if you think about it from that perspective, then maybe there's an advantage of smaller particles, and also ones that don't have cell walls that are hard to digest. There are likely differences in digestibility. And I guess I don't necessarily think about it in terms of optima, but sort of how are you getting by? So because I spent most of the talk talking about pelagic tunicates, a lot of them, the salps in particular, are really restricted to the oligotrophic open ocean. We don't see them on the shelf so much, and that's because they're really adapted to live in low nutrient water with small cells and relatively low concentrations of prey. So it's more that they're good at scooping up enough food, even if all that's there is really small things like picocyanobacteria. Interestingly, a lot of the bloom forming species, when we see blooms happening, it's at the shelf break. So I kind of think of it as like a Goldilocks situation where if there's too much food, their filtering apparatus actually gets clogged. One of my favorite paper titles had Starvation in the Midst of Plenty. It was one of Rich Harbison's papers. So they can actually starve if there's too much food. But at the shelf break, there's kind of this sweet spot where there's enough production to kind of fuel a bloom. And it's also interesting from a carbon cycling dynamic, because then at the shelf break, there's actually the potential for some of those fast-sinking fecal pellets to end up at depth where we won't see them for a very long time. How's that?
Perfect. All right, we got one more question, and I think we're going to run out of time, but I'll get up to Jessica. Meet me in the middle. Perfect.
Thanks. I'm curious about growth rate, like what we know about growth rate and how that relates to especially for the colonial ones. Are those with the most members just the oldest, or can that vary with other conditions?
Yeah, so broadly with growth rates, they can grow really fast. So one advantage of being 95 to 99% water is that you can put on size super fast. So when a solitary salp asexually buds a chain, the chain, when it breaks off, it's teeny tiny and it almost like blows up before your eyes. Kind of like those little foam things you put in water and blow up. They put on size really quickly in that way. But in terms of the age of the individuals in the chain, there's a couple different strategies. So some of them, like the linear forms, are continuous budders. So all the individual zooids in the chain are the same age, and they all kind of come off together. And they're sequential hermaphrodites. So they start off as females and then next become males. In some forms, they have, like in the cyclosalps, the buds are different ages. So the ones that came off first are older than the ones kind of trailing behind it. But in all cases, they're only living for like on the order of weeks. Yeah. So they can kind of exponentially grow when the population can grow when conditions are optimal.
Great. Any last statements for us before we wrap up, Kelly?
Well, maybe I'll just add that I have there's a current PhD student in my lab, Farzani Yasmin, who's looking at salp life history strategies. And I'm really excited about this because we kind of like we assume that all salps eat the same thing. We also kind of assume that their reproduction looks the same. And she's looking at things like the length of time, the generation times, and how many buds they're producing. And I think there'll be some really interesting insights in terms of population dynamics, because there's a bunch of unknowns there. Yeah.
Great. And I think I heard somewhere in your talk that you're willing to come back and talk to us again. So
of course, yeah.
I think that will be something we have you come back and follow up on. So that would be
great.
All right. One more round of applause for our speaker today. Thank you very much.
you.
For folks online, hope to see you again next week. Thanks, everyone. Okay, folks online, I'm going to wrap up. Thanks for being with us.